Hello, this is Eva Ansen welcoming you to this Bite-Size Bio web seminar, which today is sponsored by Leica Microsystems. Last year, Leica Microsystems launched Thunder Imagers as a new standard in fluorescence microscopy. Thunder Imagers use a method called computational clearing to eliminate the out-of-focus blur that clouds the view of thick samples when using camera-based fluorescence microscopes. We will hear more about this technology and its applications in today's presentation titled Thunder Imagers, High Performance, Versatility and Ease of Use for Your Everyday Imaging Workflows. This webinar is being presented by Dr. Falco Kruger from Leica Microsystems and Dr. Remco Megens from Ludwig Maximilians University in Munich, Germany. As always, we will have a question and answer session after the presentation. So if you have any questions, please type these into the questions box, which appears on the right-hand side of your screen, and then I will put them to our speakers at the end. The recording of this webinar will also be available online in 24 hours. So now over to you, Falco, for the presentation.
Thank you very much for this kind introduction. And hello and welcome everyone to this webcast today. That's called Thunder Imagers, High Performance, Versatility and Ease of Use for Your Imaging Workflows. My name is Falco Krüger and today I'm here with Remco Megens. Who are we? This is going to be presented. Yeah, those are the presenters of this webcast today. This is on the one hand, Dr. Remco Megens. He is PI at the Institute for Cardiovascular Prevention of the Ludwig Maximilians University in Munich. He's also head of the Advanced Optical Imaging Facility and in this position also develops imaging methods and labeling strategies for tissues in atherosclerosis research. He will also talk a little bit about the general work of the IPEK Institute later on. And this is my face so that you're also familiar with me. My name is Falco. I'm working as Advanced Workflow Specialist at Leica Microsystems. By training, I have a PhD in plant cell biology, but this doesn't mean that I cannot be amazed by any other sample. So I really love many, many different applications and specimens, and that's actually why I also love my job so much. And in my job, I'm supporting customers all over Germany, Switzerland, and Austria with trainings and guidance for their workflows. Today, we are talking about applications. So we have prepared a lot of different customer samples, so real applications from real customers and how Thunder Imagers are performing with those kinds of workflows. So it's not just about beautiful images. Today, we are going to show you a full workflow, so meaning from imaging to image analysis. So that it's also continuing and that you have a closure to your experiment. This is also something very important in the spirit of Thunder Imagers. So we hope for you actually to discover the potential of Thunder Imagers for your own applications. That's why we collected application examples from many different species and also workflow modularities. Then we will also have Remco speaking later on in the talk, who is giving a first-hand experience with a Thunder Imager that he's actually operating in his facility or in his lab. And so that we will also integrate into, let's say, more complex workflows where Thunder Imagers are actually posing as an imaging hub to streamline more advanced workflows or imaging workflows. All right, what we are not going to cover today is actually how the technical details of the Thunder Imagers work. So we will talk about computational clearing, but if you are interested in how all of the different modularities are working, please have a look at the webinar that was also recently broadcasted that is called Enhanced 3D Specimen Imaging with Computational Clearing. There, a colleague of mine, Evan Darling, is giving you the in-depth knowledge about the computational clearing. Also, there's a tech note out. So Thunder Imagers, how do they really work? I really encourage you to read this if you are interested in all the different modularities there as well. so that we can focus today on the specifics of applications and workflows. Good. So, however, to bring you, of course, to the same page, I want to start with what are thunder imagers? Thunder imagers are actually a class of wide field microscopes that are able to tackle one of the most prominent problems in wide field microscopy, and that is out of focus blur. So every time that you have a three-dimensional specimen, so let's say everything that exceeds a monolayer of cells, you have the problem of out of focus blur, because with the nature of wide field microscopes, you are illuminating your whole specimen in this field of view. So you are not only collecting the signal from your focal plane of the objective, but you're also having a lot of emitted light coming from planes above and below your focal plane. And this is, of course, then hiding details of the optical section where you're currently in with your objective. So, and thunder imagers are actually able in real time to advance the state-of-the-art fluorescence microscopy to a completely new level. And this is the three-dimensional level. So thunder imagers work in 2D and 3D. And with this actually open up so many doors in science, in life science, so that you are able here, as you can see currently, while you're recording the raw data on the right-hand side at the same time, as we call it in real time, thunder actually, or thunder imagers provide you with a really good contrast image in 3 dimensions. With thunder imagers, we are creating, or we are closing actually a gap that was long-standing between wide field and confocal point spread functions. So in the end, the diffraction pattern of light in the different microscope systems. So in a perfect world, it should be application dependent what kind of microscope you are using. However, while you have a very fast and sensitive image acquisition in wide field systems, the problem is you're, as I said before, with the out of focus blur, you're hiding your details. So most of the people are going with their life science applications to confocal systems. That is perfectly fine. However, thunder imagers are able to bridge this gap and allow you to keep the speed and sensitivity, and at the same time increase the image quality to a point where you can immediately go to data analysis, so that you can now have big fields of view with a decent image quality that allows you already to continue with your workflow. And then, for example, do real-time screening or screening in 3D specimen, so a thick biological specimen. And then if you need to go to maximize your 3D information extraction, you can go to a confocal system seamlessly and use the other Leica products like SP8 Lightning or Step 3X system. So there you can increase lateral and axial resolution even more and get the most out of your sample. So this is the general idea that gives you a huge versatility for your experiments in your everyday work life. So Thunder itself is an optodigital technology that was developed by Leica Microsystems, and the core technology here is computational clearing. Computational clearing, as I already said, removes the typical haze of wide field images because it can distinguish in real time between signal that is in focus and signal that comes from out of focus planes, and with this can actually give you really nice and contrasty images. And since this whole computational clearing is integrated into the system's data stream, you're not losing any time. So Thunder or computational clearing itself works already in 2D. No need to record 3D stacks. However, if you are recording 3D dimensional stacks, you can not only do the computational clearing on top, but we are also offering there different Thunder modalities where you can also on top have a deconvolution as well. So you're combining computational clearing with a deconvolution step that is also done with a well-known Leica decision mask technology, and this is called large volume computational clearing. So those two modes you should keep in mind. They also become important in the rest of the talk. So this computational clearing power, so the power of the Thunder imagers are actually put into a broad spectrum of systems so that we cover all the different life science applications that there are out in the field. So you can start here on the left-hand side, top left, with a stereo-biased Thunder imager, and then we go to upright systems, and down below here we have, let's say, the inverted up to the high-end systems with the Thunder imager 3D LiveSer. Why did we actually invent those kind of Thunder imagers? What is the need to actually provide you with really powerful system solutions? Well, the life sciences more and more focus on physiologically more relevant samples. This means it's perfectly fine to use, of course, mono layers or different cell cultures to do your kinds of experiments. It's perfectly fine. However, the bigger the sample gets, or the more complex the tissue gets, the more you can actually learn from those kinds of experiments. And in the last years, a very prominent sample that has come up are actually organoids. And of course, people still love and will continue to love model organisms. However, how do you want to image them without, I don't know, wasting too much time in front of the system? So the hands-on time is also something very important. So the more complex, and biology is very, very complex and most of the time three-dimensional, the more physiological relevant your samples and experiments become. Problem with the wide field systems in general is that they only can cover a very small spectrum of those kinds of life science applications. However, Thunder imagers, and that's why we have a huge family of those imagers, are invented to cover the whole spectrum. And today I'm going to show you a few examples from all of those different kinds of stages of complexity and actually also volume dimension, where you can see the performance of Thunder imagers. Okay. Before we start, I actually want to tell you, or let's say, pick your mind a little bit of how you can actually make your workflow more efficient. Let's consider a screening, for example. For a screening, you would need to image a lot of well plates and slides and dishes. Depends, of course, on the screening. So just imagine you have to screen 196 well plates or even 1,000. Or your general work day consists of you screening 100 slides for certain regions of interest and dishes and so on. So you get the idea. So usually a workflow consists of a lot of imaging that then boils down either already to your result, or if you're doing really a screening, like a chemical screen, for example, then you boil this initial phase of many, many hours of imaging down to a group of candidates or individual candidates. And once you have this, you proceed to the in-depth analysis. However, there's always also a feedback loop. So it can also be that once you have your candidates and groups, you may have to rescreen because you discover that there are more groups than intended or expected. Or once you have your first results from the in-depth analysis, that it feeds back to the kind of border or limit definition of groups. So there is a lot. So if you are doing an image-based screen, there's a lot of feedback loops and a lot of images that you actually have to acquire. And how can you make those workflows more efficient? Because if you consider only doing this on one microscope, you will clog the system pretty fast. So what we think Thunder imagers do in a very, very good way is that they reduce complexity. So to operate a Thunder imager is actually super easy. It's also a very user-friendly system in general, and also the software handling, as you know, the LASX software is very easy to handle. Thunder imagers give you a great versatility. So even though you have a single Thunder imager, you can tackle many, many different applications with those systems. And that is also very nice in terms of not needing to switch to a different workstation or to a different software that we have, of course, and not just since the Thunder imagers, we have an integrated image analysis. So you don't need to switch systems in between. The Thunder imagers, you will see this also during the presentation, have real-time processing. So this means while you're acquiring your original Z-stack, the stack that is, let's say, created on top of the raw data stack is created in real time, meaning as the images come in, you also get your computationally cleared image. So this gives you, or this drastically reduces the time to result for you. So meaning from imaging to get your final data in your hands that you can then put into a figure or something, this is drastically reduced. And this saved time you can actually use to focus on other experiments, or on the other hand, to upscale your experiments. So why don't you just use a higher N, right? A bigger area, or more wells, or more samples in general. Thunder imagers also increase your data quality and quantity. So you have the possibility to save all of those workflows as in image settings or data analysis or image analysis settings, and you can always reproduce those kinds of experiments. And this gives you the ability to have identical processing and also identical workflows even if you go months later to the system again. So you can reproduce reliably your data. And something that is really important to mention, and also something that Remco will talk about later, is that we allow that you have a multi-platform operation. So you're not only restricted to using a Thunder imager, but with our software and especially the navigator module, you can actually connect to a confocal or a stat system from Leica, the digital light sheet system, and even correlated light and electron microscopy. So there's a big versatility, and you can understand Thunder imagers as an imaging hub, where you can actually then go into the very last detail if you switch to another Leica product. And this, of course, reduces the whole problem that you have with your imaging workflows, because the problems become smaller. You get faster. It's less complicated. You get the same amount of data with just less hassle. And this, of course, then speeds up your whole workflow and allows you to become or be faster into the stage where you can go to in-depth analysis. And here already we start. So talking about shortcutting your time to image analysis. Here we have an example from Professor Mohr from the Institute for Molecular Cancer Research in Zurich, from the University of Zurich. Here on the left-hand side, you see the raw data. He's imaging cancer tissue sections that are about 40 microns in thickness. And compared to his old system where he was, let's say, used to work with a spinning disk microscope, we shortened the exposure time with our sensitive cameras down by 6 times, so 6-fold image time or exposure time reduction per section actually. And with the instant computational clearing, I already said, you get this data immediately. So you don't have to wait for this kind of image here in the middle. However, in order to quantify the individual molecules that are here labeled, so the idea behind this experiment is actually that you have labeled individual RNA molecules, then count them per cell. And to get to this kind of image quality, you have a very high-speed processing, so only within seconds in the stack, and afterwards you are able to quantify those dots, and everything while sitting in the same session actually in front of a Thunder imager. So I will also show you how the image analysis is directly integrated into the workflow. So you just need to click on the 3D button, and there you get into the 3D viewer and see actually your sample. And here on top is the analysis button, and this is where you exit, or not exit, but enter. And here on the left-hand side, then you can nicely create your image analysis pipeline for both populations. So in the end, you want to know how many spots in green and how many spots in red are there. And for each of the populations, you can create your image analysis pipeline and get to the results. And here I have a subsection where we counted actually around 950 spots in green and 542 in red or magenta. And this is here already everything done within a few minutes. Okay, and this actually saves a lot of time without, as I said before, you don't need to switch to any different workstation and come back to the system again to count the dots or anything. And with the saved time, he can now actually upscale his experiment. So instead of having a single field of view, he's now adding huge areas to his quantification. And this, of course, makes the results more and more robust. And if you are already at upscaling, what about just imaging a complete mouse retina? We have done this. The problem with a huge specimen, also they have a huge area, is that you have uneven regions, or let's say regions where you have uneven backgrounds. This can be due to the thickness of the section, that you have, let's say, out of focus, where in the end this is the problem that is hiding your signal. However, the Thunder Imager is giving you the opportunity to immediately go here with the image that you get in real time to start your quantification. You see here nicely in the line profile that you have an uneven distributed background, and once the computational clearing is done, you see here that you keep the relative intensities, but you have a very, let's say, even background, and this, in the end, it increases your contrast. Yeah, you could not distinguish the individual nuclei here by just doing a global background subtraction. So you preserve the structure, but you can immediately go to image segmentation, and this is what we have done. So here you see actually the mouse retina in the raw image file, and then now switching to the instantly computationally cleared image. See the drastic increase in contrast, and this was then giving us the right data or image quality to count the individual nuclei in the whole mouse retina sample. And just for people who are interested in that, 43,407 was the number that we counted in those kinds of retina. And this is giving you, I mean, a very nice, let's say, example. So don't stick to a single area and then just extrapolate the density of nuclei, just count them all. Another nice thing is that we, of course, have a huge and very excellent portfolio of objectives that would allow you to pre-screen also your samples. What you see here are actually explanted and fixed gonads from C. elegans. And the professor, where we have the data from, is Professor Ekman in Halle. And he's actually interested in those kinds of developments of the gonads. And he loves to, let's say, pre-screen his sections or his sample preparations, where you have several of those gonads, and then the ones that are relevant for his research, he's then going into detail and imaging them in a very nice quality and count individual cells. So here with a single shot, you get a nice overview and can even create three-dimensional volumes and select his gonads of interest in the end. What you see here is a projected image, then acquired with a tile scan with a 63x oil objective. And what Professor Ekman is interested in is, for example, the number of nuclei in this very front section here of this explant. And this is what we have done. So here again, switching to the analysis, creating an analysis pipeline that is able to distinguish the individual nuclei and then count them. And the number is actually 117. So this is another example just to integrate your analysis workflow into the imaging workflow. So going from a single field of view overview to a tile scan with a 63x oil objective, and then being able to analyze or count the individual nuclei in this section. But we can also have more complicated, let's say, analysis pipelines for your images. For example, here, it's important to quantify the cells that have a different marker. This example is from Dr. Mark Stemmler. He's working at the experimental medicine in Erlangen in Germany. And this is a breast cancer cell line that you see here. So we have actually 4 different labelings. The first one here on the left-hand side in gray is DAPI labeling. So this is used just to know, okay, where are my nuclei, and not only just bright spots on my preparation. Then we have a second marker that is guiding the scientist into the direction what kind of cell is actually worth analyzing, because a certain expression level needs to be reached in the green channel in order to be opened up for the quantification later on. So first of all, you check, okay, this is the nucleus. This is the nucleus with the right expression pattern. And afterwards, then we have 2 different markers that then are the important ones to count in the section. So again, here we integrated the whole imaging pipeline. So let's say also a more multiple step image analysis. And once you have established this, you can save those imaging pipelines and create big overviews. You can create big overviews and automatically send them to the image analysis. So this is done here. First, we selected the DAPI channel, so the individual nuclei, and then those who have the right expression level in green, and then counted the individual populations of cells. And actually, from those cells that have the right expression level, 213 of 614 were, for example, in population, the red population, and then the population 4, the magenta population, was 450 from 1,500 roughly. So this is a very robust quantification. And here you don't need to stop, okay, in your application workflow, for example. Here you can say, okay, and now I have a big field of view, but this big field of view, you could also, with the help of the LASEX navigator, upscale to a multi-well dish, okay, or yeah, there are several examples. Or why don't you go to a 96 well plate and then have those kind of big fields of view in each and all of the wells. So this is perfectly fine to do, or even upscale it more to a 384 well plates. So there you have the freedom that you need in order to have robust data. And all of this brought to you with the navigator, because there you can automatically set up those kinds of, let's say, big scale experiments very easily. It's focused on your experimental setup, and yeah, very easy to handle, because those kinds of maps here are automatically generated for you, and you just need to fill it with a certain field of view. And since we are talking about the LASEX navigator, I have here a nice example. This coming from a group in Munich. This is the group around Jan Böttcher, who is a research group leader at the Institute for Molecular Immunology and Experimental Oncology in Munich. And he's interested in he's interested in imaging as fast as possible, because he has a lot of those object slides where you have up to 12 sections. So this is only one half of the slide. And he needs to speed up his workflow because he has tons of those slides. So what we did here in a few seconds, create an overview in the navigator and then select the section that is of interest. For example, here we just used a single color and selected then the section of interest and did a multicolor overview. So in green, this is giving you the outline again of the whole section, and in red and blue, there are different kinds of cells or nuclei marked. And once he has this multicolor overview, he selects actually the points of interest that he needs to image in order to get his quantification that he needs. And what you see here is the raw image projection of a Z-stack, where you can have a look at the instantly available Thunder image that has already very nice quality to it. So in order to segment, you could go even a step further. So use the large volume computational clearing, where you add the deconvolution on top and create an even better image quality in order to count those kind of cells. And you can do all of this even with multiple colors without losing any time here. So this is still from Jan Böttcher. He is using up to 6 different colors. He has an example with 5 different colors where you can nicely see the individual cell types, and also that he's able to distinguish between those different cells. If this is still not enough, what he can do is, and what he actually does is that he uses his SP8 Falcon system, and there with a white light laser is actually the spectral freedom, and can also add a whole new contrast and the contrast of lifetime to those multiple color samples, which is actually really good. Another thing that you could do is actually go with those kinds of samples into the cryo pipeline. So why don't you do a correlated light and electron microscopy with that? Here also we have a Thunder Imager application workflow where you can use the power of Thunder Imagers and integrate it into your cryo light microscopy workflow. Okay, so far I've shown you a few examples already for, let's say, smaller samples, although they had sometimes a big area to cover. But now we are going 3-dimensional. So, as I stated in the beginning, to the more physiologically relevant examples or specimen. So here you see a piece of a mouse intestine that was imaged together with Dr. Selina Kepler. She's also a group leader at the Center for Translational Cancer Research in Munich. And just have a look at the dimensions. So here we have a piece of organ that is 2.5 by 2.5 millimeter and 1.5 millimeters in depth. And you can actually see through this kind of cleared piece of organ. Labeled here are the blood vessels in magenta and the epithelium in green. And this whole overview was actually recorded with an air objective. Okay, so with a 10x air objective, with a tire scan, a 2x2. And you can see the image quality that is outstanding in order to focus down what kind of region of interest in this tissue or organ section is relevant in order to go to a multiphoton system, for example, and then make really in-depth imaging. What you see here is the 3D overview of quite a big organoid sample. We imaged this during a workshop in Erlangen where Atria, a PhD student at the local university clinic, brought this impressive 3x4 millimeter uncleared human brain organoid. And within only 7 minutes, we created an overview of this whole specimen and acquiring a 3x4 tile scan with 4 different color channels in over 210 microns of depth. And I think you will agree with me when I say that the quality is more than sufficient to pinpoint individual regions of interest that then are used for later analysis. And this is another human brain organoid, again uncleared. So actually the biggest workflow efficiency increase for Atria was that she doesn't need to clear her organoids anymore. This doesn't mean that with Thunder Imagers you don't need to clear any organoids or samples anymore, but just test it and see what the Thunder Imagers can do for you. Here you can, for example, see that this overview just took 68 seconds to be acquired, and then the additional processing with background removal, with the computational clearing and the deconvolution on top only took a little bit more than 2 minutes. You can actually sit in front of the system and see how the different deconvolution settings impact the quality of your stack. And I think it's a very nice example. Here she was actually interested in this area here on the left-hand side where this human brain organoid actually proliferated already to a more complex structure. And then we went for the 20x long working distance objective, so to get a higher magnification. You see also how the image quality or the stack quality improves. And we are still air objective, so we could actually also image those kinds of samples through the bottom of a well plate. Okay, so far we only use the 10x and the 20x long working distance objective, which allows you to do this. For higher resolution, we then used the 20x plan APO objective, and here you can see how the data was increased drastically in quality, and you can see the individual microglia cells, how they move into the tissue. And this is actually what she was interested about, how those or how many of those cells go how deep into the tissue. Another, let's say, group of specimen are, of course, the model organisms. And my secret passion, plant cell biology, is also part of today's webinar. And here you see on the left-hand side the raw image of an early endosome marker that was actually provided by Professor Karin Schumacher at the Center for Organism Studies in Heidelberg. Here you can also see how efficiently Thunder Imagers create really nice, crisp images. And what can you do with those nice and crisp images? You could do endosome tracking, for example, because this image quality is quite nice. Here, for example, you see a 4D imaging example. So I projected the volume that was acquired over time into a single plane. What you see here is the movie of the epidermal cell layer and the moving endosomes in between. You can really nicely see the streaming of those endosomes. And those, of course, those particles you can track and actually measure the individual velocities of those endosomes. Yeah, you have a few of them just wobbling around, but then you have some here that really move in a very, very fast way. But Thunder Imagers are not only very potent in plant cell biology, but also plant developmental biology. What you see here is a growing root tip that was imaged over several hours. You see how the root grows from left to right. Yeah, here's actually the root tip, and those what is actually labeled are nuclei, and you see here that those nuclei that are moving outwards are actually root hairs that are projecting from the developing root. And yeah, it actually took several hours to record this movie because the root is growing over a millimeter during this time-lapse movie. And then, of course, you can track individual nuclei and see how they developed over time or how the cells divided over time. This is not the same root, but a similar specimen is here a large field of view that was projected. So here we recorded a multiple tile scan in the Z range as well, and projected the image, and you can see here over 7 millimeters the root tip here in the front, where you have expression of a certain cell marker, and then cover the nuclei over the whole root. This is a close-up to the root tip, but a different one, where you can see the expression of different cell markers. Okay, here actually the interesting topic here is the quiescent center region, where it's the stem cell niche of those root tips. So you can, with a very sensitive cameras that we offer with the THUNDER imagers, you can get a really nice result or to analyze the expression pattern. However, if this shouldn't be sufficient, you can still move, let's say, use the THUNDER imagers to pre-screen all of those roots that are relevant for in-depth imaging, and then move, for example, to the digital light sheet microscope that also is offered by Leica. And here you see an example of actually one of those root tips where you can then nicely segment those kind of nuclei. A very nice example here is also coming from plant physiology. So it's again from the Center for Organism Studies. This time Rainer Ward was providing the sample here. And what you see is actually true multiplexing in Arabidopsis roots. You have the top 2 rows a ratiometric sensor that are giving you the consumption of ATP in those kind of cells where it's expressed, and then down below that is very dark is an intensiometric calcium sensor. And this, of course, I mean, if there is calcium flooded into the cytoplasm, then of course you get an increase in intensity, and this is what is measured. And here on the right hand side you see how robust this kind of measurement is. This is recorded with a time interval of 6 seconds. So this is quite decent, and this you also know from your for example, from a confocal system where you also want to do this kind of measurements. However, the true beauty of this whole experiment was actually something else, and this is the true field of view, because I was hiding a little bit of detail from you up till now. This was the real field of view that was covered in 6 seconds, so 3 channels, and you can actually now follow a real calcium wave through the root. Okay, it's not only an area where you see the calcium levels rising and going down again. Now you can really follow from the trigger points that we set here that there's a calcium wave moving through the whole root. Okay, so here you can follow it again and see how quick it is, but still you can resolve this in a time-wise manner. But why stick only to the root? Here you see a whole Arabidopsis seedling where calcium waves is going through the root first and then moving over. Here you see how the speed decreases. However, the calcium signal must have been transmitted via other structures already, because you can see that there are also calcium waves in the, happening in the cotyledons. It's one of the very nice examples from plant physiology. So don't be afraid to use THUNDER imagers. They can cover a huge area really quick and in a very sensitive way. I will play it again once. You can enjoy it. All right, and I think for now I am done, and I will soon hand over to Remco now. I just want to make the point again that the combination of THUNDER imagers with the other Leica portfolio is actually rocking your application workflow. So you can combine Lightning and Thunder. This is actually something that you should do. You're very welcome. Just contact your local account managers and ask for a demo or a quote. But just to make a point again, Thunder Imagers offer here the solution for you, that you can now decide really what is the importance of your experiment. What is the question you want to answer, and what kind of system you are going to use for it is what kind of resolution you need. Do you need the speed component, or do you need the actual resolution component? And then you decide, okay, it's a Thunder Imager or it's an SB8 confocal. And with this, I'm handing over to Remco. Thank you.
Thanks, Falco, for the introduction. And I'll continue by shortly introducing myself again. So this is me. Falco already introduced me. I work at IPEK and also have an affiliation in Cardiovascular Research Institute in Maastricht. And a little bit more of history on the institute I work for, also to get some background on what I will show later. So it's an institute for cardiovascular prevention, and it utilizes a lot of microscopy to mainly study cardiovascular and immunological targets. And at present, we have several conventional and also several advanced microscopic imaging systems available here. So amongst others, a multiphoton system, a confocal/STAT system, and nowadays also the new Thunder Tissue Imager 3D. Here you see some examples of what we've been doing in recent years. So we focus on whole mount ex vivo and in vivo. Here you see some examples of bone marrow. Here you can see recordings, a variety of recordings of large arteries. We study a lot of atherosclerosis here as well. But we also do in vitro cell culture, cell isolation in high magnification and lower magnification. And of course, we do a lot of histology as well, which you can see here. So why a Thunder microscope at IPEK? But we would like to see an increased imaging capacity. So that's why we thought we need another microscope, especially for immunohistochemistry. We would also like to improve the quality. So with the same effort, getting a better picture out of it, that would be ideal. And also expand the possibilities we have. So more 3D and potentially also 3D cell culture. The ease of use is very important. This is a clinical/scientific institute. So not many people are really interested in tuning and playing around with microscopes. They should simply work. And overall, I think it's always important to improve science. So with a Thunder microscope, we thought we could use thicker sample size and also faster and easier overviews that would help our understanding of the target. So another reason was that we would also consider Thunder to be a partial replacement of our confocal system, which is quite well booked and sometimes overbooked. So maybe the simple confocal questions could also be solved with the Thunder. And with that, also the workflow of the institute should be improved. And finally, we were hoping, or that would be really great, a great bonus, that also the 3D capabilities would also help us in whole mount tissue imaging, with or without clearing. And on the right side, you can see the Thunder imager we have in the institute. I simply took a picture of that. It's quite a small footprint, so it's nice to fit in any lab. So this Leica Thunder webinar from my side is important to demonstrate the correlative power of Thunder within the advanced confocal microscopy and nanoscopy. So what I mean with that is that we would try to use the platform of Thunder to make it more easy to work with these more high magnification systems. With that, we should also, again, improve workflow for some experiments, and also improve the quality of the data. So if you can spend more time optimizing your picture rather than searching it, that's always better. And of course, the whole mount 3D tissue would also save us a lot of time if we can do that with Thunder. Our target sample of today is the Omentum. And the Omentum is taken from a mouse. It's a Seek3CL1 GFP reporter mouse in this case. So what is the Omentum? The Omentum is a visceral adipose tissue sheet in the abdominal area of the mouse. And it contains also some lymphoid structures called fat-associated lipid clusters, or in an abbreviation called FALCs. Here you can see on the right side, you can see a schematic overview of it, where you can see the Omentum in gray, that's adipose tissue, and you can see in magenta the microcirculation, in blue you see the lymphoid circulation, and in green we can see these nodes, these FARCs available. And these FARCs are involved in immunity, so they play an important role there, also in disease, but also in steady state. And the number of and size of these FARCs does change upon inflammation by recruitment of lymphocytes and macrophages. But also, the Omentum has a very nice microcirculatory network, and which is why I actually chose this sample for this webinar. So what will we do? We have a mouse Omentum. We will mount the fixed Omentum on glass, cover it with a correct cover slip. We've used ImmuMount to mount it, and it's not clear. So this is simply not fresh, but it's simple, normal tissue. And then we will use the THUNDER microscope with a fluorescence microscopy mode. We will use a 10x, as Falco already mentioned, that's a really nice objective. And we will use the navigator function of LAS-X. So here you see the first example. This is raw data. It's a maximum intensity projection of a large overview. And this is really large, so it's like almost 7 millimeters in length. This was taken relatively quickly, much quicker than with any other microscope we have in our institute. And on the left, you can see the number of nuclei. This is a simple Hoef staining. In the middle, you can see the GFP of the CH3CL1 positive cells. And on the right hand, we can see the microcirculation, which is labeled with a CD31 that labels endothelial junctions of the normal vasculature. If you put all these layers together, you can get an overlay picture, and that you can see here, where you can still see all the structures, but it's a bit blurry because of the thickness of the sample, which is approximately 150 micron, so rather thick for a normal microscope. And if you then look a bit more closely into the raw data, you can see that all the structures are clearly visible, but also the blur and the cloud is covering a lot of area. And the nice thing with Thunder is if you then apply that, which is really instantly, all of a sudden you get a much clearer view. You already can see it's much more crisp. The vessels are much more distinctively visible. But also, if you look at these FARCs, these green clusters, I mean, you can really count single cells now there. So that's absolutely a great improvement. And it's also a standardized improvement. So the machine does the work, meaning that the user can really, it's not really involved in that. That's good for long-term stability of data. There we go. Here we see it in a movie where we just removed the nuclei, the DNA staining for ease of visibility. And you can see small clusters of GFP combined with these microvessels. So the next thing I would like to show you, besides the quality improvement of the Thunder system, is also the integration. So you can use it as a correlative tool to go to the next platform. And in our hands, that will be a Leica SP8 stat system. Which is right in the same room. So why? Well, if you have a high magnification, you will also, in the end, get a much higher resolution. So the Thunder system is really good, and we've seen from Falco that you even can get high resolution quality as well if you put other objectives on it. Since we have a big system, an advanced system, which will even give us a better result, for us it's really interesting to make this correlative step. But why don't we do it directly on the SP8? Well, it's very difficult to find your target in a large tissue sample. Which area is interesting? That's really difficult to decide if you only see a very small field of view. And also the laser scanning based systems are intrinsically slow, at least they're slower than a camera based system. We do have a resonance scanning system, which is already much faster than traditional scan head, but still it does not meet the speed of the Thunder system. So the nice thing is that if you have two systems of the same company with using the same software, you can actually use the same data set to go and apply that to the other system. This is the system I'm talking about. It's the SP8 3X. It's an inverted stand with a white light laser. It has internal hybrid detectors. And in this case, we will use high-end, high magnification objectives, 63 or 93 times. It has a navigator software as well as the Lightning package on it. And it's again in the same room, so that makes life really easy. Here you see a screenshot of the LESIX navigator of the SP8, where we did some scanning, and here you can see the field of view, and we did a spiral scan, which is there in the left bottom corner, to basically detect the sample and to study the sample where we are. As you can see, we did quite some of the sample. You see a lot of cells. That's the spiral scan overview. And what we now want to do is actually go with the right mouse and load and align another image. So we will use the overview made by the THUNDER microscope in the navigator of the SP8. Here we are. So here you get another window where you can select your picture, which we will do. And then the align image window will open. And you can see on the left side the current picture, or the current area on the microscope, on the SP8 microscope. And on the right side, you see the imported THUNDER overview. It's actually the same one as what we've seen before in the movie. And it is of note that if you switch from an upright, which we have for THUNDER, to an inverted, you may have to flip the picture, which you can do on the left-hand corner, lower corner of the window. Then we zoom in both pictures to get more detail, and we try to find an area where we can say, okay, this is the exact same area where we are. It's not always easy, but very often at the edges of the sample, there's a couple of typical structures which you could be able to use to track your position. In this case, it worked. We found a couple of cells which are really typical. For example, at these blue spots, you can see cells in a typical alignment which are the same. So we're quite convinced this is the good area. Then you can go to the next step, and you can actually create reference points. You have to take two locations in the picture to enable the calibration of the overview with the live mode picture. And that you can see here. I zoom in a little bit more to make it a bit more clear. You can see where the arrows are, the two spots, which are selected on the reference image, the live image, as well as on the overview THUNDER image. And once you're happy with the alignment, and the more accurately you do that, the better it is, you press align. And that's it. It's quite simple. Then you come back to your navigator window. It's the acquisition window, and you see the overview combined with your current field of view and your spiral scan area. And then all of a sudden you start to notice that your spiral scan, which looked quite big, is actually rather small when compared to the whole overview obtained with THUNDER. And I think this is the key of the THUNDER microscope in this correlative or platform shifting science or imaging. That small white box, in this case now made yellow with an arrow, is the field of view. It's the actual field of view, and this is, I think, with a 93 times objective, so really high magnification objective. It's very small, and that's the spiral scan area. So if we zoom in a bit closer on the image of the THUNDER system, you can draw a box, basically an area of interest, with the navigator. There we have the field of view again, and that area will be then scanned later on. If we zoom in a bit more to this box, you can see that navigator calculated with the settings of the zoom, the internal zoom, as well as the objective, how many fields of view you would have to image. Even more close, actually it turned out we need 6 fields of view to study this area in total. Also, again, really powerful tool to have a navigator on a system with high magnification systems, objectives. Once you're happy with the area, you select your desired image parameters and you start the sequence. And then as a result, you get a couple of really nice pictures. As you can see here, here we see the microcirculation. This is the CD31, and you can see that we have a lot of detail. Of course, the quality with an SP8 system and really good objectives is very high. If we then add the green channels, or the GFP positive cells, which are mainly macrophages in this tissue, and the nuclei, you can see that you get a lot of additional information on top of the overview information. So basically, by combining the two microscopic systems, we really go from one tissue area or the whole mount tissue studying the relationship between areas and microcirculation, to highly detailed information where we can see single junctional areas for endothelium cells, for example. Of course, we can also make movies out of that. And here you can see a movie of this area where we also apply the lightning and we remove the nuclei, and then all of a sudden you also can see that these green cells are surrounding microvessels quite often. You can quantify this, so that's a big step forward in quality and in detail, but it really is an add-on to the THUNDER system. So beyond seeing what is, which is not possible with an SP8, THUNDER adds that, and on the other hand, the SP8 adds the super high quality, which THUNDER is possible to achieve, but will take more effort. All right, lightning is the next step that really also improves. I'll play the movie again. Here you see it without lightning, and then you can see the crispness of the nuclei becoming much more apparent and much more clean. Okay. Then we move on to a final slide. Not yet, actually. So in conclusion, we can see the overview with THUNDER, and at the same sample, in the same experiment, in the same room, we can actually add the high detail or the high magnification information. So it's a really cool combination and very powerful for the workflow in our institute. So in conclusion, the THUNDER imager in our institute is really also a workhorse. It's not shown in this webinar, but it's really important just for bright field and fluorescence images. And it can also replace the simple confocal experiments. So we can free time to do the more advanced sessions only on the SP8 and not blocking a lot of time with the simple ones, which could be done by THUNDER. That also improves the workflow in our institute, not only by on a daily basis by having more time on each system, but also overview images, which usually take a lot of time with our other microscopes in our institute, can be done much faster. So that's absolutely a big positive. The improved image quality. I could have never imagined a couple of years ago that with wide field you could obtain a whole mount Omentum in full detail. That's really impressive. And the 3D capabilities does make even more experiments possible. So it's actually enlarging the possibilities we have and studying things. And I can imagine that cleared samples, for example, could be really good on our THUNDER system. And it's a multi-platform tool, so it really mixes up well with all the other systems we have. The software is basically the same software as on an SP8 or on a multi-photon system we have, so people can switch relatively easy between all these different technologies, which is a very important factor in our institute. Okay, this is more or less what I had to say. I would like to thank Leica also for the opportunity to talk, and I would also like to give the word back to Falco.
All right, thank you, Remco. That was a really nice story again, and this is also why I have here again the slide open. That what Remco just showed was really the movement from top left to bottom right, That he uses all the different modalities that Leica offers in his microscope portfolio and use it to get from a big organ overview to individual cells and then see really the last intricate details. And this is really something super nice, and that's also why we gladly invited him to join us here today. Okay, so wrapping up this webcast of today, I hope we could show you that THUNDER imagers are really an amazing advancement in wide field microscopy that allow you to streamline your workflows. THUNDER imagers can be applied to many different workflows, specimens. You have seen tissues, individual cells, you have seen organs, you have seen Arabidopsis seedlings that were imaged in total, And all of this really with scientific meaning. We showed you quantifications and how data analysis integrated in the THUNDER imagers as well. And all of this was intended to demonstrate to you that you don't need to be afraid anymore from really a big amount of imaging that you have to do in your lab. Now you have the Thunder imagers at your side, and this really can largely, or you can actually largely benefit from those kinds of imagers and yeah, advance your science as well. And with this, I would like to thank you. And also, I would like now to hand over again to the question and answer session. Thank you very much.
Thank you, Falco and Remco. That was an excellent presentation. We have a few minutes now to address some of the questions that came in from the audience during your talk. If anyone else has a question, feel free to post it in the questions box that appears on the right. And if there is time, we might ask it, but there's quite a lot of questions to go through already. First, a question about the image of the whole retina that Falco showed as an upscaling example. Did you use stitch image there? And if so, how many times does it take to acquire the whole retina?
Yes, thank you for the question. So indeed, we of course used a tile scan experiment here. So I think it was about 90 individual tiles, so individual positions that were then stitched to the final image. And the time it took was about 14 minutes. So what you need to know about this retina section is that you also need to acquire a small Z-stack around each focal position, that you make sure that you really get all of the different nuclei, because they're not all of the sitting in the same height. And yeah, so it's a small Z-stack at 90 tiles with 2 colors in about 14 minutes.
And we have a few other questions that were about people's specific research organisms. And you'll like the first one because it's about plant science. From your experience, what is the maximum acquisition depth in Z that still gives a good resolution without pre-clearing for plant imaging?
Yeah, without knowing what kind of specimen it is directly, or I'm hesitating to give a strict number. I would say as something between 100 and 200 microns should work easily. I mean, you have seen root specimen that were not cleared. I mean, the root tissue is in general a very clear tissue, and it's readily symmetric, and we have been imaging around the quiescent center. You saw those kind of experiments there, and so you can easily image the whole root if you want. And going more upwards, the shoot, cotyledons, we did already several experiments, and there you can easily go through the epidermal cell layer, right? I mean, here the power is really that you could, like with the Omentum, create a 3-dimensional overview of a cotyledon if you need, and then really see also their differential expression of, let's say, developmental marker proteins or something. So I would, in general, I would say just check the LinkedIn webpage or the webpage of Leica itself or the Twitter channel, and check where the next workshop of Thunder Imagers is, and then just bring your sample and have a look yourself.
And of course, not everyone is working with plants. So we also got a question asking, can we use this technique to study and analyze all forms of microorganisms like bacteria and viruses?
I would, so it depends also on the workflow, or let's say on the experiment itself and the question you are asking. So we have been working with scientists, for example, that work with bacteria, with soil bacteria, and let them grow within, I don't know, a 3D printed mesh of wells. And then those wells are connected with small tubes. And then in the end, they want to see how bacterial population 1 moves towards population 2 and so on. So if it's about quantifying or tracking individual movements of bacteria, this should be fine. However, if you want to study substructures of bacteria or even viruses, this is not possible. I mean, is it still a wide field microscope? I'm happy that people are so amazed that they also think that they can work with viruses there. But the point is, detecting individual virus particles and bacteria is perfectly doable. You can also quantify that. We also did this in tissue sections already. However, if you, and this is what I try to point out with this point spread function overview, if you really need to extract the highest resolution information, then you need to go to a confocal or stat system. Yeah.
We've got a few other questions, and I think we have a few more minutes. One other question that came in was, is it possible to do quantitative analysis with Thunder processed images?
Yeah. First of all, in general, what is quantification? I mean, all the different workflows that we showed today were about quantification. So we quantified information that was within the data sets. So yes, you can quantify Thunder data sets, but I think that the question is more towards quantification of intensities. And here, working with wide field microscopes is always quite tricky to do absolute intensity quantification, because you always have an overlap or an overlay of background light and the specific signal that comes from your structure of interest, for example. And from my point of view, I would say, I mean, what we do with the computational clearing is that we remove the unevenly distributed component of background in your images, and what is left are the structures and the signal that come from the in-focal plane. Okay, and here I would say you can, or you should actually use only the instantly computationally cleared images, because they're lacking the background. Okay, and I mean, the absolute intensities are not the same as in the original image. However, the relative intensities stay intact, and we're not changing anything from the structures here. It's not deconvolution. Computational clearing is removing the background. And here, I think you can be sure that you can also quantify those kind of instantly computationally cleared images.
Okay. Yeah. And someone else would like to know if there is a possibility to add multiphoton feature to the Thunder microscope.
So multiphoton excitation is a confocal technique or a confocal illumination. And we also not planning to do this on wide field microscopes. So yeah, this is more a short answer, and it's a no.
I've got two more questions of the ones that came in the last few minutes. There's one that a lot of people will find interesting. It's about research publication. Some research journals want to know which algorithm is used for image processing. So what published algorithms are working in Thunder, and where can I find the publication for that?
This is what I told you in the beginning, so that we have a tech note out, where application specialists and also product manager, let's say, did several different experiments to really show that the Thunder algorithm is working as intended and as we say it's working. And there are also more in-depth explanations on how the calculations are done. Yeah, and this is also, I mean, it's online, it's available, so you can download it and have a read, and maybe then also make sure for yourself that if you like it or not.
Yeah, and I've posted a link to the tech note in the chat window, and there's also now a link to Thunder Image Gallery in there. There's one final question that goes into a little bit of technical detail. It is, why is it not possible to use bright field light Z-stack scanning with Thunder, and is it only possible for distinct wavelengths?
So,
a hard one to close.
The computational clearing is currently, as we offer it, is a technique that deals with fluorescent images. So here, I mean, in the end, you need to specify what kind of light you are using. So the light that comes or that is emitted from your sample has a certain wavelength. And if you just, I mean, in the algorithm, we are using also the properties of light, and we need to know what kind of wavelength is working there. And then, of course, you need specific information about this. If you now would use a bright field image, and I'm assuming again that the question is more regarding a color bright field image, so for example of an histological stain, then you have an overlay of many, many different wavelengths, and you would need to distinguish that. And currently the chips that record those kinds of light information are separated into red, green, and blue. Okay, and those are only 3 wavelengths, or let's say in those bands they are detecting the light, and this is then not very, let's say, scientific to do any processing on those images. This is more like, if the question is, will there be thunder also for bright field images? I would say you would need to wait for this a little bit longer.
Okay, well, thank you very much. This brings us to the end of the seminar. So thanks again, Falco and Remco, for a very illuminating presentation and a great discussion. Thank you to the sponsor. You're welcome. Thank you to our sponsor, Leica Microsystems. And finally, thanks to all of you, the audience, for taking the time to attend and listen in. If you have enjoyed the seminar and would like to view the video recording, please visit the seminar page on bitesizebio.com, and it should be on there within the next 24 hours. And there you can also see other webinars we have lined up for you. So until next time, good luck in your research, and goodbye from all of us at Leica Microsystems and Bitesize Bio.